Hi, everyone. Thank you for joining us today for our fourth SynBio and control theory breakout discussion. So far, we've had three really engaging ones. This breakout is going to be focused on scaling biocontrol methods to complex systems from cells to tissues. We have two hosts, just like our previous sessions. Professor Marcela Gomez, she is from the Applied Mathematics Department at UC Santa Cruz, and her background is in mechanical engineering. Her research interests are synthetic and systems biology. Also hosting is Dr. Eric Johnson, who is a program director at the National Cancer Institute at NIH. His interests cover automated microfluidics, tissue engineering, and single cell approaches. So with that, I'll pass the virtual mic to Marcela and Eric for their introduction and to start the breakout.
Great, thank you, Jerma. And thank you, everyone. Thanks, Eric. Right, so as Jerma mentioned, this breakout is focused on scaling up biocontrol methods to complex biological systems. So we'll briefly provide an outline of today. So we'll begin with just a short background for motivation. I'll begin by discussing a case study for existing work in feedback control of complex biological processes and all the challenges that are contained within that work, followed by Jerma, who will give an overview of work in cancer and open challenges where feedback can help. And then we'll briefly go over the agenda, a short icebreaker so that we can all get to know each other, and instructions for our breakout activities. So before we begin, we're going to share a link. So we were thinking about how we might be able to collect participant information. And this might be useful either so you can see who else is in the room, if you want to build collaborations. And so these are all the links are going to be to Jamboard. And so on this one, if you click on the link there that Eric has kindly provided, to the left, there's a place where you can add a sticky note. You can place it on the board in real time, and everybody can see what you post there. And so the sticky note that's already there is mine. And so what I've added is my name, some institutional information, what is my current work, or why am I here and what am I interested in? So for me, for example, I've been working in one domain application. I'd be interested in working in other domain applications. Additionally, the location of the sticky note indicates basically your background and training. So are you more of an experimental researcher? Are you more mathematical and computational? Are you somewhere in between? So this way we can start to organize ourselves accordingly. So, feel free to do this during our introductory talks, and then eventually we'll revisit it and see where we stand. And let us know if you have any technical issues with that. Okay, so with that, I'll begin the introduction, the motivation talks. So I'm going to talk a little bit about approaches and challenges in the control of complex biological processes, but within the context of my work. And I'll preface by saying that, we're not the only, I'm not the only one thinking about this work, but simply using it as a guiding mechanism for this discussion. And so the focus of our lab has really been on thinking about control at the extracellular level. So moving away from genetic regulation, single cell level engineering, but rather leveraging the fact that, that we can control cellular response by manipulating the environment around it, right? So, but if we can understand how cells respond to changes in the environment, then essentially we can potentially direct cellular response by intelligently manipulating the environment accordingly to achieve the desired behavior. And so through this mechanism, tight spatial temporal control will allow us to enhance current developments in synthetic biology. And so examples include smart bandages. It can also be stem cell differentiation, control, and patterning. And this is relevant for applications such as limb regeneration and meat culturing. So if we continue with this premise that we can potentially direct cellular response or biological system response by manipulating the environment. And we want to achieve a desired behavior, then the question is, what is the right stimulus or changes that we should introduce to the cell in order to get the desired output that we want? And can we do this in an automated way that's precise and predictable? And so in that light, what can an engineering approach do for our understanding? Because if we can achieve that, then presumably we can also gain some understanding in that process. And so Richard Feynman famously said, "What I cannot create, I do not understand." And in some sense, we're here to challenge that notion. What does it mean to understand? There are different levels of understanding, and what can be gained from each level, right? And how much understanding is enough to move forward? For example, engineers, we create all the time without sufficient understanding. So an example of that is synthetic biology. So what is, like I said, what is sufficient understanding? Is sufficient understanding achieved when we can control a system? And so current work has already showed capabilities of implementing feedback control by coupling sensors and actuators in order to direct cellular response. And so examples of these have been like controlling stem cell membrane potential, with the goal of controlling stem cell differentiation, controlling cell migration. And so these are areas we would like to develop. And so in this example, we have the controller is external to the cell system. We have a culture here under a microscope. So the information that's being received, that's sensing the response of the system, is a microscope. So this is mostly image-based. Those images can then get processed into a feedback control algorithm, which then can manipulate the extracellular environment, thereby modifying the readout of the system. And so in our work, in my collaborations, we've been working with developmental cell biologists and also those that work in bioelectronic devices. And so this is an example on the left of a bioelectronic, this is a proton pump, for example. So we can culture the cells on top of it, apply differential voltages, and this can deliver or remove protons, thereby changing the pH of the environment. And so we are able to use this to control the readout of stem cell membrane potential. And so other researchers have done similar setups where we have this external feedback. And so if the input to output response of these systems are predictive and reliable and either consistently positively correlated or negatively correlated, I always expect that an increase in the pH is going to increase the fluorescence readout or vice versa. Then this control scheme actually is very simple to achieve. But what begins to complicate things is when we start to get nonlinearities in saturations in our system. So that means that when it gets a little bit complicated, where the input to output response relationship or correlation is no longer preserved and dependent on the input. So we've recently been working on moving this framework to wound healing. So the same setup where you have a bioelectronic device that can deliver and remove ions, ionically charged molecules. So we can deliver therapeutic molecules. We can also apply electric fields, for example. And again, we've been limited to imaging. So the information that we receive of response to the system once we deliver these things is again through a camera. And so Eric will touch on, essentially the challenges and limitations and how we can think beyond just the imaging component. And so as I was saying, like here now we have a more, sorry, go back. So now we have here a more complex system, right? Where we have this we have a coordinated sequence of biological processes that must happen in order to achieve the wound closure. So if we think about how do we, in the last slide, we were thinking about feedback control. It's a very simple input to output response system that we're regulating. But in this case, we have a response that not only scales spatially, but also scales temporally And when you're intervening in this feedback control, then it's going to depend on at what time point in the wound healing stage you're intervening and which biological process you're targeting. And also with the knowledge that there can be off-target effects and that you're affecting other biological processes at the same time. And so reading out also the information creates a challenge of, when you do actuate on the system, how do you get it back? How do you measure the response of the system? And so in this particular project, our goal is to reduce wound healing time by 50%. With intervention strategies that include reduced time of inflammation by manipulating the macrophage recruitment activation, and accelerate proliferation stage by increasing innervation rates and directionality. And so some of the control challenges that we have been facing is, one is like guaranteeing safety. So we really just been thinking about acute wound healing, not thinking about chronicity or how we can avoid perhaps going into a state of chronicity or perhaps even cancer if we're accelerating proliferation, for example. The set of reachable states are unknown. So reachability in control theory is this concept of like, if a system is reachable, you can go from, point A. There's basically a way to get from point A to point B, guaranteed, right? You find the right stimulus that's going to get you from one point to the other. If it's not reachable, but it's possible that it's not reachable, that you may not be able to achieve a certain state of the biological system. Or you may not be able to, or there's no guarantee that you'll be able to find the path to reach that state. Also, system response is highly variable and can change if the intervention strategies are too forceful. Meaning, we tend to think if the input to my system is some signal A, then I should always get out signal B. But that's not true, because it's possible that the system adapts, a new pathway upregulates, and then you input signal A. And then perhaps, an hour later, a day later, you get signal C out instead of signal A. So that's another challenge that we face in biology. Again, going back to limited observable states, which was really forced us to infer dynamic relationships across these spatial temporal scales. And so far it's been a lot of inference just using qualitative model and the limited data that we've had access to. Also few and small data sets. So, we can do microarray analysis. That's something around 30,000 genes, but within those 30,000 genes, we only found a handful to be predictive. And furthermore, these handful can be clustered into 5 states. So really the amount of information that's contained in there does not correlate with the dimension of the data set. And then we need a lot of temporal information. So dynamics is key in understanding how to do real-time feedback control and response to a system. And most data sets, their time series data can be short or sparse in the temporal resolution as well. And finally, we have limited capabilities, biophysical and biochemical actuators and sensors. And this is something that, we would like to take advantage of this meeting to figure out more solutions for, and Eric will speak more to that as well. And so Eric, I'll hand it off to you now. Thank you.
Thank you, Marcella. Yeah, so just to kind of reiterate what Marcella touched on is trying to actuate beyond imaging. A lot of the times we work with some of these automated microfluidics, we're kind of combined with looking at the cells, looking at molecular changes via fluorescence, updating our systems based on that imaging output, and then trying to cycle that back in. And just to give you, a quick kind of view of my experiences, working with E. coli, cell sorting with microbiome, methanogens, trying to do this all in planar extensional flow, and really trying to incorporate, some of this automated solutions using LabVIEW, controllers, and then even the hardware has gotten advanced, and now we're scaling up to even microwell arrays, microwells, co-culture platforms, organoids on chip. So it's really nice to see is how can we leverage that, and some of the projects we've had coming out of the Physical Sciences Oncology Network, and really trying to integrate physical approaches to cancer research, has been this cancer tissue engineering collaborative, really trying to develop and characterize tissue engineer technologies to reflect the pathology and physiology of human disease, and how this specifically relates to cancer. How can we utilize this to couple with common cancer models? And then how do we build upon this with these multidisciplinary teams? And then really interfacing this back with some of our larger programs across NIH. And so what that looks like in terms of the challenges is looking at these complex systems and their feedback response. So as I mentioned, multi-organ on a chip. How can we couple, whether it's vasculature, immune components, bone niches, and being able to look at how primary tumor sites can metastasize into secondary sites, trying to isolate, circulating tumor cells, looking at the models between intravasation and extravasation. How can we combine that in some of these systems? Coupling in these immune responses and how these changes relates to the tumor microenvironment. And in doing this with the synthetic biology approaches, are there new materials that can help kind of couple all these components together that we have yet to explore in terms of other fields such as engineering or material science? And they're really trying to, to thread this balance between reactive, proactive versus predictive. We have all these different timescales identifying these changes. Is there ways of identifying these molecular single cell changes, immune response? If we focus on one, do we negate the others? And how is there better ways of doing this? And then, beyond creating models that are better for basic research, alternatives to animal models, being able to couple these organs on the chips, can these be robust enough for these adaptive kind of control mechanisms for patient specificity? And can these controls be modular? So finding ways, and we talked about in some of our last breakouts, some of these standards and frameworks. Are there ways to couple in these frameworks together to build these kind of modular-based control adaptive systems? And where does synthetic biology help to meet that versus the automated component? Finding ways to where the modeling, the math can help us, but really the actual incentives, as Marcella pointed out, whether these are molecular changes, can these be read out beyond imaging? And something we were really looking for in our discussions today. So with that said, I just want to close with this last slide. We all have these problems within our fields, and we all have the one field that wants to come in and solve the problem, but realizing that this is a very complex problem and it runs, very diverse approaches. And so you can decide on what you want to put there instead of algorithms, but in reality, I think combining, as we're all aware of combining all our approaches, sharing with each other these challenges. And one of the things we realize is we all want to have databases or libraries or things that we can explore with, but we'd really like to hear is what you want to do with those. If you were to have, one of these advances or new materials or technologies, what would you actually do with it? And I think in sharing those visions and what you have and what questions you want to ask, within the room, you'll realize there's others who maybe have solved that question and used a different approach. So really trying to drive at some of these finer barriers and making sure, we're utilizing kind of the knowledge in the space that we have. So with that, so today's timeline, we're going to have topic, after we do the icebreaker, we'll have topic 1. And then we'll do that for 45 minutes. And that's going to be a Jamboard, and we'll talk about that in a little bit. And then we'll follow by topic 2. And then we'll all come back together into the main room for our last remaining topic 3. And then I'm going to hand this over to Marcella to just quickly describe this breakout Jamboard structure.
All right, thanks. So yeah, we designed these Jamboards to facilitate discussion. So it looks like we'll be doing 2 breakouts with the given group that we have. So if we'll provide the link for this, and if you click on it, I think there's breakout group 1, 2, 3, but just go to the one that corresponds to your breakout group number. And so you can see there are prompts for the designated spaces on the Jamboard that are delineated by some shape and color. And there are some sticky notes, so you can just double click on the sticky notes, add your notes as you're in the discussion. So if you don't have an opportunity to vocalize any of your ideas, you can also just put them down here. So we'd like to fill this up as much as possible. It would be great if we ran out of sticky notes, and if you did, you can go to the left and you can add additional sticky notes. So you're always welcome to drag in more. We just kind of preset these just to make it easier. And on the top left, there's usually some example discussion points that you can jump off of. And so we'll do this for topic 1 and topic 2. We'll do the breakout rooms. And you'll go out to breakout rooms for a little bit, and then we'll come back for a collective discussion. So depending on how juicy the conversations are in the breakout rooms, we might keep them going a little bit longer just to not interrupt the flow. But when we come back, we're going to summarize essentially summarize the discussion, the major discussion points. So please do identify somebody who will report out for your group. And then topic 3, I guess we'll revisit this again, but in topic 3, we'll do that all together in one room.
Great. Okay, so I'm going to post this link again in the chat, and then I'm going to pull it up real quickly. Let's see. Okay, I just posted the link again for those who may have joined late. And it's great. It's great to see that. Let's see, we'll see what this looks like real quick. Perfect.
This is like 3 clearly delineated groups here.
There you go. Great. Yeah, continue to add that for those who just joined. I posted the link in the chat, and we'll revisit this again towards the end, but this is great. Okay, and now I'm going to switch quick over to the last thing for this intro. So I would like, while I pull up the slides, I would like everybody to grab a piece of paper and a pen. It could be a sticky note, whatever you have in your vicinity. Let's see. Let me share my screen. Okay, so what we're going to do is for this, quickly, we're going to do this kind of icebreaker to really get us all on the same page. We're really looking at how this demonstrates effective communication and goal setting. All right, so with your piece of paper and your pen in hand, this will be quick. You're gonna follow the instructions on the next slide, and good luck. All right. Does everybody have a paper and pen? All right. Okay, so first draw a rectangle. Okay. Next, draw a large reverse letter C that sits at an angle on top of that rectangle. Okay. Next, draw a square at the top end of that C you just created. So I'm just gonna pause here. If everybody can raise your hand that you've got to at least these first 3 instructions. I want to make sure everybody's able to follow along and see if there's any issues. So as a show of hands, everybody ready to move on to the next instruction? Okay, great. Thank you all for your responsiveness. Okay. All right. Okay. Draw 2 more smaller rectangles at the lower end of that square. And draw a smaller rectangle or smaller flat rectangle at the top end of that square. Okay, I think this is the last instruction. At the lower end of the C, draw a rectangle that is parallel to your first rectangle. Okay, drew what they drew. Would anybody like to show what they drew? Hey, Ross. Hey, Ross.
Sure, I'll start.
Yeah, what do you got?
How does
All
this
right.
look?
That looks good. Okay. What do you think that is?
I don't know, 3D printer or something like that.
All right, anybody else? That's one data point. And we got a second data point.
We got Peter.
It's hard for me to see. Go ahead. Let's see.
Looks
I'm gonna quit the screen share so we can kind of see. There we go.
Yeah.
Awesome. Okay. Cool.
And no idea what that could be.
Anybody take a guess at what it could be? One word description. Okay. I'll pull up the answer. Let me. Okay. Can anybody tell what that looks like? So it's good. We had
I'd say Ross was pretty close. Mine
Yeah,
was way off. That's for sure.
Okay, so what I like doing about this activity, this is great, thank you all for humoring us. What this really shows is that we've all seen and heard the same set of instructions, but we all interpret them very widely, right? So as we communicate with our colleagues during this breakout, we have to recognize our different perspectives and approaches and how we communicate our interests and our challenges and how we're overcoming them. The other thing, what I like showing about this is without really setting the goal or the vision of what you're building, you can get varying different results or applications. So, as we talked about in past breakouts, knowing in some regards, there's a lot of a benefit of having this basic exploratory research. And on the other side of that, for those with the engineering mind, they like having that application, right? So how do we bridge that divide to be able to do both? Because I think both are important, both in terms of the basic, basic research and how that pushes these advancements, and then from the translational engineering approaches, how can we, get to that? So I appreciate you all. Thank you all for the comments, and feel free to use this for your own breakouts. Okay. All right, so now we're gonna quickly jump in. I know it's already 12:35. Do you want to open this up, Marcella?
Yeah, so we're going to kick it off with topic 1, challenges in generating predictive models for biological systems, slows the design of complex systems. So this is really just founded on, I feel like that has been one of the largest barriers, is our inability to come up with predictive models for biological systems. So that really slows this sort of like iterative feedback process of designing systems. And perhaps is like just limiting overall in the time scale at which we can advance. And so the prompt is, can robust control be used to engineer system response without a predictive mechanistic model? Like, is our only hope that we can build a predictive model from the ground up, or are there ways around it? So this is going back to what is sufficient understanding in order to achieve our goals in engineering, and bioengineering in particular. And so Eric will provide the link shortly for topic 1 Jamboard. And so I said, well, we'll go into 2 breakout rooms. And then I guess Eric and I will each be in one. since we'll have two, that makes sense. And yeah, so then we'll continue the discussions in the smaller group. So hopefully everybody feels a little more comfortable speaking up. And then we'll come back to the large group. And so again, that's about 15 minutes we'll convene, depending on how the conversations are going. So shall
Great.
we?
Yeah, I just put the link in the chat. Make sure, does anybody have any issues accessing that Jamboard? So this will be Jamboard Breakout Topic 1, and we'll have 2 groups. So once you become assigned in your breakout group, just go to that. And at the top, you'll see in the Jamboard, on the very top, there's arrows left and right. You can cycle through the different Jamboards, as Marcella mentioned. Okay, does anybody have any questions? Okay, great. We're looking forward to discussion.
Hi, can you see me? Yes, here I am. How are you, Marcella?
I'm good. Good to see you. Good to see your name. Oh, we
Good
could
to
do
see
a round of introductions probably, since I don't know everybody.
Do you want me to start?
Yeah, sure.
Sure. Hi, everyone. My name is Anya Baitika, and I am an incoming assistant professor at Drexel University in Mechanical Engineering, and my background is in synthetic biology and control theory. I work on designing biological feedback controllers.
And we'll popcorn it over to Peter.
Hi, I'm Pete Carr. I'm a senior staff scientist at a part of MIT called Lincoln Laboratory. That focuses especially on national security questions and applications. And I'm particularly interested in biocontrol related to biocontainment of engineered organisms.
Thanks. Ross?
I'm Ross. I'm a postdoctoral fellow at the University of British Columbia. I work on designing feedback and feed-forward control systems for mammalian cells and programming development of stem cells to immune cells like T cells.
Well, thank you. Paiksu?
Hey, I'm Taesok Moon. I'm the WashU professor, but today I'm representing EBRC, Engineering Biology Research Consortium. That is the nonprofit organization to provide synthetic biology solution to global problems, to global community and White House and basically all the funders in US. So nice to meet you. I'm working on synthetic biology too long, almost 2 decades, and my interest is also too many, but mostly biocontainment and probiotic. If I talking about NIH related one.
Thank you. And Tuba?
Hi, Marcella. I'm Tuba Fier. I'm a program officer at the National Institute of Child Health and Human Development at the NIH. I am not a synthetic biologist by training, but I'm a geneticist, cell biologist, and developmental biologist. So I'm really interested in the intersection of synthetic biology and its applications to the study of embryonic development, for example. So looking forward to the discussions.
Great. Thank you. Jermont, do you want to introduce, or I guess you did already.
I can just say a little bit about my interests. So I'm a program officer for the National Institute of Biomedical Imaging and Bioengineering. And what we like to do is look at tech dev. So tech dev doesn't have to be something like a device. It can also be a synthetic biology component. So the overall kind of program that I'm envisioning is engineering biology. So it'll encompass pretty much tools, techniques, processes, models, and along with everything that's kind of being discussed. I actually spent lots of time at Hanscom, so I know Lincoln Labs pretty well. That's interesting, and I know plenty of people from EBRC. So this is a nice confluence of all my history.
Okay, I'm gonna see if we can you guys see the Jamboard? Okay, so this is our slide. So it'll say breakout group 1. So do you guys all have the link? Yeah, okay.
Yes.
Okay. Well, let me know if you weren't able to get it before we were pulled into our rooms. So this, I mean, this is essentially where everybody can just feel free to speak up, but these were just some example discussion points that I had popped into there, that I hope that by no means should be limiting our discussion. But, you know, in thinking about ways to circumvent the need for predictive mechanistic models, you know, the first thing you might think about is data-driven models, right? And so if we are to use data-driven models, you know, what are the limitations and concerns for such an approach? The other question is like, do we firmly believe we can get away from mechanistic models altogether, or, you know, or do we feel like convicted that they're absolutely necessary, and that perhaps what we should be focusing on are ways to accelerate the development of those predictive mechanistic models? So, you know, I'm not trying to say that one way or the other. And then another major concern is that if we are thinking about biomedical applications and the like, how can we ensure safety, right? And so that's more like on the ethical side, since now it seems that more and more there's mandates to consider the ethics of the research that we're doing, which comes through sometimes in some of the funding announcement opportunities. So I think that should also be, you know, within this discussion. So we can jump around. We can begin with the left. What's the latest and greatest? Or you guys can just openly share. And if you just do so, just your thoughts, like, you know, what are your feelings in general about this area? Maybe it's a good way to open up the discussion. And I'm happy to also fill in some of those notes as we're talking.
So can I start?
Yes.
So I strongly believe, you know, for engineering or predictive model-based circuit building for many, many years, more than a decade. And at the same time, I also try to, I mean, actually, I'm constantly every day asking what is the governing rules in biology, as I mentioned in previous the session. But today I kind of I'll be thinking more, you know, different direction after the exercise we just did, drawing exercise. So this is kind of question to everybody, because I kind of immediately feel something a little bit different direction. That is basically, let's say we have somehow luckily whatever governing rules in biology or predictive model somehow, and each researcher interpret them models, also use them model a little bit subjective or different way. And then what would be consequence of that application of model to individual experiment? That was kind of one thing I came to my mind. And then if we apply the same, you know, rules or models in a different way, there might be, I don't know, there might be, I don't know, I mean, this model might not be useful, or still useful, but we will generate too many different outcomes. So that kind of, after exercise, kind of thinking about that one, and then that kind of occupied me. So I just want to hear from other people.
So to summarize that, I put a couple of sticky notes on your behalf, which you're welcome to edit or add. So on the latest and greatest, I put, you know, model-based circuit design and search for governing rules for that. And then can a mechanistic model hold in all contexts? I think what you're saying is that, you know, we could spend a lot of time building up a mechanistic model, but in unseen environments, does that, you know, that model can break down, right? And so then the question is like, how much more effort and time and money is it to then adapt that model or build a new model? Hi, Tuba. You had to see your hand up. Sorry.
Yes, so I have some thoughts about that. I think it's really important to put the context around what you're trying to accomplish. I think, based on the exercise that we had, if we all had a context that this was going to be a microscope, we may have come up with different designs of the microscope, but at least we had the idea that it was going to be a microscope. So that really helps define the rules, because there won't be any single rule that applies to every context. And I can, excuse me, give examples from developmental biology and the development of the embryo. Same signaling molecules are used over and over again, but based on the tissue or the timing of development, outcomes are different. So it's the same signaling molecule, but the outcomes will be different based on context. So you really need to first start with the confines of the problem and the context you're working under, and then evolve your tools as you change context, because the rules will not hold as context changes. So I really would want to emphasize that.
I think that's really great, because I think more broadly, just in the field of biology, right? I think the hardest thing for me was when I started going into biology, and especially into biomedical stuff, or it just seemed like there was always papers that had conflicting answers. Like when we started doing the wound healing, we were trying to understand what were the biomarkers for macrophages, and that was a huge rabbit hole. And I never imagined it would be, and different papers listing different biomarkers, and sometimes they were in one list and not the other list. And it was the context, right? The context in which they did the experiment, the context of the conditions, the context of their starting cell line. So more broadly speaking, would it help if the fields agreed on a common context for us at least to begin advancing uniformly in one direction, and then we can, with that understanding, be able to expand out. So I added a sticky note for you there, but feel free to add more.
So Cuba, I like your answer, but I want to ask another question, because that's my entire 20 years of question. I still don't have answer to that. So I also completely agree with you, your point regarding context dependence, everything kind of rely on those contexts, because that is my experience for last 20 years. But at the same time, I try to discover governing rule that are applicable to every single living system. Of course, we have the one that is the central dogma, and the other is evolution. And do you think we could find another governing rule that applicable to entire biological system? Like physics, we have, whatever quantum mechanics, principles. Chemistry, we know thermodynamics rules. Will we have those kind of rules, governing rules, or we cannot?
I don't know, that's an interesting point, but I would argue for because you mentioned central dogma. So central dogma can only do so much for you when you want to interpret these rules, right? I think one thing I would argue is that epigenetics is a big component there. I think that's as close as you can get to a governing rule in living systems, is the context of epigenetics, because the DNA can only tell you so much. It tells you what a cell can become, but it does not tell you what it will become, right? In the context of differentiation, development, and whatnot, because is the cell capable of becoming something else based on its chromatin context, epigenetic context? Because the DNA is the same, but how it's packaged and whether it's available for interpretation to make decisions is a different story. So it's interesting. I would still argue that even central dogma is not a general enough principle to really explain everything.
Add that.
One thing I'm thinking about is starting at the lowest level, would it be possible to fully model predictively, mechanistically, whatever, one gene? So taking into account everything we know about gene regulation, maybe in a bacteria or mammalian cell, whatever, try to understand how different context effects can affect that one gene. Synthetic genes are relatively simple. They don't have as complex of regulation as endogenous genes, but you could choose ones that are sort of increasingly complicated in their regulation and try to build some sort of model. Because if you try to start with the whole system at once, I think that you're going to run into this like overcomplication issue, basically. But if you can start with something small and try to fully understand that, especially if it's very important to some system of interest, then maybe that's a way to peruse the question of how much can we predict about something. And if you can basically try to build a model, whether it's mechanistic or not, that accounts for context dependence for that one gene, then you could at least see if it's feasible and to what degree or how much detail you need, how much data you need to be predictive.
I've seen some work in that area, and I think maybe that's what made me lose a lot of hope, is that I've seen people do that on a plasmid, like facing one direction, facing another direction, next to the origin replication, away from the origin replication, changing the RBS site, the promoter site. And all these led to wildly different things. And, there's some labs that they're in, the entire 30-year career is building this database of promoters and characterizing predictive promoters. So it is one approach, but then I guess the question is like the timescale at which we advance in that approach. And we're trying to achieve these like higher level things like stem cell differentiation patterning and maybe directing embryo development. When are those going to meet? Or are they just completely two separate research areas that we need to think about completely different, right? And I also want to jump us off to, because we're probably going to end the breakout room soon, moving that to the orange box that is like, what is lacking in methods for control in math? And I think the problem there is that we have all these nice mathematical formulations, and guess what? We require like these massive amounts of data that would cost millions of dollars to produce, right? Because we want time series, we want dynamics, we want different inputs, we want to see what happens to the system, we want to span the entire dynamic exploration state. But that's impossible too, right? We can't do that approach as well. So like if you can think about like on the mathematical side, where can we meet in the middle? What do you wish that like if you thought that math and machine learning was black magic, what do you wish it can address with what you have? Or what would you like to see it do?
Yeah, so to second what Marcella and Ross were saying, I mean, there's like labs that just build a predictive model of a single protein. And they look at it in all the different states that the protein can be in, and there's massive models, and they take lots and lots of measurements of them, and they're highly predictive. So that's one end. And then the other end is what Marcella was saying, people have started moving towards machine learning and building surrogate models. But the problem with that is you need data to train them on. And the question is how much data and how expensive it is.
I guess I would argue that if you're doing a data-driven approach, right, you're sort of limited to the data in. So how do you predict context that you haven't seen? With the mechanistic model, I guess in theory, also, you wouldn't necessarily build a mechanistic model that incorporates context that you haven't seen either. Although feasibly, like if you have a model of some signaling pathway, let's say, and you just build it up to say, okay, how do these like 5 proteins interact with each other? What are the feasible interactions? Then even if you haven't seen some context where they're interacting in a certain way, it could capture that behavior. So I guess how do you bring that together? How do you use the data that is out there to sort of seed, maybe not like a fully mechanistic model, but something that captures the emergent properties of the system so that feasibly it could reproduce some sort of responses that are not in the data.
So I added for that, mechanistic models capture emergent properties, but I would say not predictive, right? And then, and feel free to add notes. And the data-driven methods can extrapolate to can data-driven methods extrapolate to context? Is that possible? Can we design such a thing? Okay. Yes. I see Suba's on the top, so I'm guessing it's the order in which you raise your hand.
Yeah, I'll quickly chime in. So I'm not a computational biologist. I don't know how these models are constructed, but I think of genetics and, gene interactions. When people construct their models, do they weight their models? Because when I think of, protein interactions, gene interactions, not everything has equal weight, right? We think of as geneticists, we think of, is it necessary? Is it sufficient? Is it a cofactor? Is it a component? There are factors that you absolutely require for the process to happen. And there are others that are sort of co-players. So they don't have equal weight in making those decisions. So do those models take into account the relative weight of the effect size of these nodes?
That's really
mentioned.
That's really great idea. Relative weights of effect size of nodes. Sorry, I got to write this down. Yeah. There may be, not that I'm not as fully explored, and I really like, just based on your comment, I really see the importance of it. Go ahead, Pete.
So I was wondering, with respect to our question for this topic, can robust control be used without a mechanistic model? The one option that's been put forward is data-driven, sort of machine learning oriented methods as an alternative. I'm just wondering, brainstorm-wise, are there any other alternative categories that come to mind beyond that?
Yeah, because yeah, that's true. Because I think that we're kind of stuck in either mechanistic versus machine learning, right? But is there something in between? Can we leverage both somehow?
I mean, you can use feedback controllers to try to minimize some of the lack of knowledge of the predictive controllers, of the predictive models, right? That's what the engineers do.
There's also physics-informed machine learning where you constrain the system in some way based on
that's right,
how gene regulation can even theoretically work. So for example, like somebody in our lab, TM, who's in one of the other breakout rooms, has been looking at this as a way to sort of collect a bunch of data, for example, from sequencing, port that over into some generated model of a gene regulatory network. So nothing crazy, just looking at some transcription factors that interplay with each other. And you constrain the machine learning to try to determine the parameters of like a simple transcription factor interaction network. And so it somewhat bridges the mechanistic to the machine learning, but it's neither fully mechanistic because it's still pretty high level and phenotypic, but it's not completely unconstrained either. So I think that is building upon those kinds of approaches might be useful.
Yeah, that's an interesting point. But I also want to ask the question to everybody, because let me put it this way. I am a big fan of the mechanistic model, but now I orbit more toward machine learning based, modeling. The reason is what? So let me put it this way. We don't have any governing rule in biology except, I would say, the evolution and central dogma or something. To me, whatever mechanistic model we currently have basically made using 100 years of biological data we have. So that is sort of machine learning model, right? Based model in a sense, because we collected data for 100 years and then, or whatever decade, and then we build whatever mechanical model that's not completely describe entire process because we don't know all the details in biology because biology is too messy. So having said that, what is really the mechanical model? because at the end of the day, that mechanical model also based on all the data we collected, several decades. So now, so that after I realizing that one, now I, rely on more machine learning based model. And actually, we also working on one model, machine learning based, prediction model, and then that is working very well somehow. And then, so I mean, that's kind of my question to everybody. What do you think? I mean, the point I just made.
Kuba, you have a response, or is it different? Heading in a
Yeah,
different.
it is related to that. So I had a comment, but I think it feeds into what Taisok said, in that I think there's real value in using machine learning, but I don't see it as sort of meeting in the middle. I see it as sort of feeding, like, going back and forth, feeding and learning from each other and refining the model. So you use machine learning to extract information about the mechanism, then you make better predictions, and then you go back and adjust your models. But one other application I could see with, and this ties into what Ross said about gene regulatory networks, just because transcription factors are at the same place at the same time doesn't mean they interact. They're part of a gene regulatory network. Everything is timing dependent, chromatin effects and whatnot. So I could really see machine learning methods to not only to point at what's going on, but to point at what it could not be. You know what I mean? So if there is a way, based on all the information that we have, the machine learning can tell you these factors are not involved in this process, just by elimination, going by elimination, to reduce the possibility space, that then you can maybe look at more mechanistic relationships, if that makes sense. So that's one way of looking at it.
Thank you.
Yeah, something related to that. Thanks for bringing up a good point too, but I saw some interesting work. I think it was by James Sharp at in Barcelona. I forget what institute there, but one of the EU institutes, I think. And what they were looking at was sort of, I think, limb development. And they built an ensemble of models and then tried to figure out which ones couldn't explain the data, right? And so it was strictly based on the architecture of some gene regulatory network. And so I think that this idea of trying to eliminate possibilities through like machine learning or ensemble approaches is like a really interesting way, especially for like natural systems, to understand how they're working. And then from there, with the limited space, should be more easy to either build a mechanistic model or build a predictive model in some way, whether data-driven or not.
I like this process of elimination that you both brought up. Anya, we're gonna like be pulled out in 1 minute. Go ahead.
Sure. I was going to say people already do process of elimination with mechanistic models and have been doing it for a while. I mean, that's basically what we do when we write one down implicitly, or you can do it more explicitly and more rigorously. But also, just because I'm not a specialist in machine learning, do machine learning models actually give you the possibility to eliminate models?
Not that I know of, but I thought it was like such a great idea, because I'm also thinking about like what can machine learning, what developments should we focus on in the advancements of machine learning approaches, and you guys are all bringing up really great ideas. And I think interpretability of machine learning has just become a hot topic, so that's also something that we could not get before. Some information about mechanism that we can iterate on. At least the algorithm may do something or inform something, but we would have no idea why. Really, or a fundamental understanding.
Okay. Yeah, and also, does it actually go back to inform your mechanistic models? Because it's just like the computer doing a linear whatever under the hood.
My limited understanding is that there are some modes of machine learning that can inform on mechanistic models.
Okay.
Cool. Okay, you guys add all these notes in the Jamboard. Also, we'll continue this conversation in the main room, so we're all gonna report back and summarize.
approaches, what is lacking in methods for control in any mathematical approaches, and then what questions remain to be explored. So if you guys, I know, we have some time to kind of go through this. We can, if you want, why don't we start by just introducing yourselves, your experience and your interests in joining today. Would that work?
Yeah, sounds great.
I'm happy to start. My name is Uduak George. I'm an assistant professor at San Diego State University, and I'm in the math department. And what I do, I use mathematical modeling to study tissue development. And right now I'm looking at lung development and mammary gland development.
I can go. Let's see next.
Okay.
Welcome and thanks. Thank you so much for organizing this. I'm Ákos Nyerges and I'm zooming in from Harvard Medical School from Boston. I'm working on synthetic genomics, synthesize whole genomes for exciting purposes, for example, biocontainment, to be able to prevent horizontal gene transfer, both incoming and outgoing horizontal gene transfer, and develop and produce drugs. Yeah, I'm happy to chat about this. More
Great.
during this session.
Thank you both. I'll go since I'm the third one on camera. As you all know, I'm Eric Johnson Chavarria. I'm a program director at the National Cancer Institute. So I work in the physical sciences and oncology space. And as I mentioned, Cancer Nutrition and Genomics Collaborative. So looking forward to discussions. And I think we have on the call in our breakout room, we have, and apologies with my names here. We have Tiam, Julia, and Lee. Would you guys like to just quickly introduce yourself and your interests?
Yeah, sure. Hi, everyone. Sorry, my camera malfunctioned this couple of days, so it's gonna stay off, but I'm Tiam Haidari. I'm from UBC. So my background is in physics and biophysics, but I moved to bioengineering and working with the stem cells for the past few years. And I'm working around using synthetic biology and mathematical modeling and biophysical modeling to first study and then guide the collective or multicellular dynamics of stem cell driven tissues.
Great, thank you.
So I'll go next. Hi, everyone. I'm Julia Ringel. I'm a health specialist at the National Institute for Biomedical Imaging and Bioengineering. So along with Jermont Chen and the rest of the Synthetic Biology Consortium at NIH, I've been helping organize these breakouts as well as next week's overall consortium meeting. So thanks for attending.
Thanks, Julia. We can see you, we can't hear you, Lee.
But yeah, I'll go briefly. My name is Lee Sims. I'm an assistant program director at the NIBIB, one of Jermont's colleagues. I'm just here taking notes in the background. My specialty is in biomaterials, slightly different flavor, but thanks.
Thanks, Lee. Okay. So I guess between Ákos, Uduak, and Tiam, what do you guys believe is, or I guess, what would you like to highlight as the latest and greatest in the space? You know, what are some of the things you've seen that have been really, catching your interest right now? Whether it's on the synthetic biology side or the control theory approaches or both. Curious to know what is really, driving kind of some of your interest in your research. And if you want, feel free to put that on a sticky, or you can say it and I can do my best to add it as well.
Yeah, maybe I can start first. So because I'm working with the stem cells, and probably most of you know, stem cells are great at interpreting inputs and basically dynamic inputs into outputs. Basically, they see signals and mechanical cues or chemical cues, and then they translate it into changed physical, like basically gene expression state, and also their physical properties. One thing that I notice is exactly the problem that you're mentioning here. how much we need to model something based on the mechanism that we know, like mechanistic modeling, to be able to control it. And one thing that I like recently, and I basically caught my attention more and more, is what is the intermediate scale that we can model biological systems that will still have some information about how to control them. But also we don't need to really go to all the details and nuts and bolts of what are actually the specific mechanisms happening. What I'm basically focusing on, I like more and more, is treating the cells at some sort of black boxes that process information, but still yet have some predictive model of what would be the output of this information processing unit, and try to alter the signals instead of going down and trying to change the gene networks or gene expressions. Is at least the tool that I'm trying to use. And maybe it goes back to some works from Thomas Gregor, William Bialek in Princeton, that they try to somehow relate the input of signals or morphogenes into gene expression without modeling them, with more than 99% accuracy.
Thank you.
Yeah, it was a great summary, Tiam. I only have a few things to add. So we also becoming better and better in predicting failure modes and how like biological systems are constantly evolving, especially if you're trying to control them or trying to pose selection pressures or try to express things that cells dislike. And yeah, the things we are trying to edit, engineer, control are constantly responding to these inputs and what we are doing to them. And we actually became better in understanding how these evolutionary processes then impact synthetic circuits, how they impact the functionalities that we are trying to achieve. And by understanding these, in one hand, the field itself came up with better strategies to engineer cells in a fail-safe manner, try to pose less, have less impact on cellular processes, cellular resources. We heard great summaries about these in the previous meetings, a week ago, 2 weeks ago. And I think the field is moving towards having robust control tools to, even without understanding the full cellular system and how these cells respond to what we are doing to them, to be able to control them and modify them in a less failure-prone manner. So, for example, genetic code engineering. That's why I'm really excited about these technologies, is because these are, in many cases, unbreakable evolutionary gaps or unbreakable evolutionary steps, and cells can't really transverse them with single or simple mutations. So if we have more tools, redundant CRISPR systems, redundant transcription factors, which can provide some redundancy, we can force the cells to do what we want, even when cells really dislike the modifications or the things we want to achieve. So we can become better in doing these and understanding what the cells will respond.
So it sounds like you're putting up like guardrails, really trying to either focus them or scope them into a certain evolution.
Yes, exactly.
Yeah, I think it's a great point, because maybe I can add to, at least in the context of mammalian and developmental biology that I'm working on now, is one issue that we have with that is these systems are really complex in the terms that are evolutionary, basically evolved to be robust against perturbations, and there are multiple layers of compensatory mechanisms that exist there. And if we want to tweak A, B comes and basically try to cancel whatever we are doing. And recently I was thinking that maybe can we really try to decouple the system completely from its input signal and then try to manipulate it from that perspective? Might be and shorter term solution than actually understanding everything. Particularly when the system grows into a multicellular system, for example, as it happens in embryo or a tissue of cells. Cells actually divide the labor, and when a group of cells do something that is not supposed to, the other ones try to compensate or send signals to actually get them back to track. And this is actually a challenge for us, particularly because we are looking at multiple cells. So I don't know what your perspective will be there in terms of evolution. It's another nice point though.
Yes, absolutely. Mammalian cells and eukaryotic cells are much more complex, and we don't really, or previously, like while for bacterial systems, we are at a stage field-wise where we can resynthesize genomes, remove regions. We kind of have an understanding of what does what, even if it's not all the time correct, and there are still black areas. But in mammalian genomes, it's exponentially more. So maybe not today, but in a few decades from now, we will also have the same tools for mammalian genomes, and we will have the same capabilities or very similar understanding in bigger genomes as well. And we might be able to achieve the same level of control, what we can do with smaller, simpler prokaryotic genomes. I think it's just a matter of time, tool development, and a lot of effort to reach the same level with more complex cells and organisms as well. It's an exciting period of synthetic biology to be in.
Yeah.
We will see these in the next 2 decades.
Yeah, absolutely.
So I'm going to turn over to Udak. So I like your background in terms of the physical approaches. So you have some background in fluid dynamics, mathematical modeling, and I was really interested to see how, recent papers in terms of tumor microenvironment stiffness, viscosity really impacts, cellular migration. So beyond looking at, we'll have a lot of, genetic readouts, really, at least selfishly from my portfolio, we're really looking at physical approaches and how we can utilize that in terms of average centers to really kind of, whether or not you want to put these guardrails on this evolution to really able to study it. And for this kind of basic cancer research. So I'd love to hear your thoughts in terms of your experience.
Okay, in terms of my experience, I'm going to be talking about, based on what you've explained, I think sometimes that in the mathematical modeling realm, we don't really get the data that we need. You biologists might think that they have put out all the data, but sometimes there is a problem where we have the mathematicians going to their own conferences, and then the biologists going to their conferences. And then we are working on the same problems, but then we are working at it in different ways and using different tools. And I think most of us have to come more together in the sense that we all have to be able to have a way where data is available. If, let's say, for example, we start going more to the same conferences with the biologists, the models will be better tailored to be able to answer specific questions. And if we are all building models, I know there are some models in my field that we look at, we say, this is phenomenal. But then you see a biologist come in and say, we know this already, then why is this being built? So I think that it would be a lot more progress being done if we have this convergence of people from different disciplines. It's happening a lot, like in my own case, I've been collaborating more with my colleagues here in toxicologists, and being able to get data has helped us to kind of like have PhD students which we mentor together, and understanding what's going on in their field helps us to make better contributions. And looking at groups that have been very successful, like some groups out here at UCI, they've done a lot of work in modeling and also contributing to how the data that is being obtained from labs in the biological people in biology, how we can better develop models that can help. I think that if we have those types of groups, which a lot are being formed, And I'm seeing so many of those things coming out now, where you have models that are tailored to better answer specific questions. And if we that are doing mechanistic models keep doing our own mechanistic models based on what we read in the literature without actually talking to people that are working on that, then we are not going to do a good job. So that's why I'm here today, to kind of like learn a little bit more about what the other groups are doing in terms of people who are not basically in mechanistic modeling like me, to see how I can understand a little bit more about what are the open questions and what are the needs, so that I can better tailor my models in that direction.
Thanks for that. Thanks for that perspective, because you raised something that I'm kind of actually interested too. So one of the things in my past, being a former training director, and we talked about a lot with our cancer systems biology consortiums, our AI model consortiums, before it was, we really needed the data scientists, and they would learn the biology as they went along. And now, because we have these models, we have these approaches, and some of them are, straight off the shelf and can be integrated pretty quickly. Now there seems to be a shift in that you really need to have kind of this hybrid training between understanding the complex, modeling, but as well as understanding the biology to know and to ask the right questions. And so I'm curious from you all on the call here, how do you think in terms of, training, what would be needed to kind of get at some of these topic questions we're proposing today, having these really highly integrated training components, focusing more back on the biology to make sure we're asking the right questions, or is there ways we can communicate our approaches, both in synthetic biology engineering aspect versus whether it's biology on a specific health or disease, or as you mentioned, you raised like you're working with your toxicologists, your colleagues across the hall. What have you all seen in terms of your institutions, the kind of the things that should be really implemented more broadly to really get at some of these challenges?
Well, maybe I can just expand, answer your question in terms of in my institution. We have a Biomat group that has been here even before I joined. And with the colleague Biomat, where we have colleagues from biology and colleagues from mathematics that work together weekly. And this has been running for over 15 years. And I was just right before I came here, and they've been hugely successful because of having them working and talking together and working on joint project and joint mentoring students. And that has really worked in our field. And I started out mentoring an undergrad with my colleague in toxicology. From there, we were able to, mentoring a PhD student together. And having a PhD student, a mathematics PhD student, going into her lab, learning how to do the experiment that she works on with zebrafish, learning how to use the microscope, and also learning the computational skills that she needs to be able to understand how to read the data. And now, since she's a PhD student, she's able to explain to me what my colleague is talking about, because sometimes the language is different. She could say, she talks about differentiation. It means a different thing to me as a mathematician versus her talking about cell differentiation. Having a student in between who goes to her lab and talks to me as my student helps me to better understand what's going on, explaining her findings to me, and then we can better model. So that's the two different ways that I've seen it being successful.
Thank you. Akos, Tim, do you have any thoughts?
Well, it is probably somehow related to one of the slides that I think it was you or Marcella that showed around this famous quote from Rick Feynman's that basically what I can build, I can understand. And I think it is quite of a coincidence, because maybe like the past couple of months, I'm thinking seriously about this. And the more that I'm thinking about it, I'm becoming more close to this conclusion that it is probably this code is coming from a different context and not for necessarily biology. because it looks like for biology, because it is mainly emergent properties are out there, the map of mechanism to function is not one-to-one. Sorry, my cat is here screaming. Because of that, I'm not sure, even though when we build something with the similar function, we necessarily converge on the same type of mechanisms. And that's quite of a challenge.
Thanks.
Yeah.
Akers, do you have anything to add? And then I was going to quickly jump to another thread.
Yeah, thanks. My view, or how I see these, is that in biology, we are becoming better and better in understanding biological properties. And indeed, we see that what we design doesn't function as we design. There's a contextual variation, cell-to-cell variation, et cetera, et cetera. And these are functions that we can't currently model. But it doesn't mean that we might not be able to model these or understand these model these biological features, phenomenon later on, maybe in 50 years. While in physics, we kind of already understand chemistry, we understand much more, we have a better understanding of how these processes happen, what are the factors that influence what we see. In biology, we are just trying to decipher and understand these now. We slowly but surely have the computational capacity to model biological processes. And I think in 50 years, a few decades, we will be able to much better predict the biological processes that we can only observe right now. So I think we are getting there. It's just a lot of scale up, standardized measurements, more and more measurements to try to understand all the sequence space and to be able to extrapolate from the measurements we have to actually predict biological functions and sequence to phenotype relationships.
Thank you for that. So we have about 5 minutes remaining. What I would love to do, can you all see the Jamboard? Are you all familiar with Jamboard? If you can just take a moment and put in one sticky, and they could be big or small. It could be what really comes to mind, your gut, your experience, just putting something down in terms of the latest and greatest that you think. I try to highlight what we were talking about in some of these. Feel free to add one. If you can do, 2 of these, great. In one or, there's 4 subsections here. If you can do 1 in all, that's great. And I'll just use that time to kind of put something down, and then we can come back to it at the next one. Does that work?
Yes.
Okay. So if you double click on a sticky, it'll pop up and you should be able to type it in and hit enter. That way we can capture some of the thoughts you all raised and kind of the great comments you made. Okay, I like it. So continue to add. We have about 50 seconds left, and then we'll be put into the next room. And then we'll kind of share what we've kind of come up with. But these are great, and I appreciate all the comments made. We'll do this another round for the topic 2. So.
All right, I think we're all popping back in now. So we're going to have, we had some really interesting discussions. So hopefully we can continue them collectively, just presuming that perhaps the discussions might have diverged in each of the breakout rooms. Yeah, would somebody like to summarize from breakout room 1 from our group? Anybody, any brave souls? High level.
Okay, if no one's going to do it, I'll do it. Okay. I think that discussion centered on a few things. So the first and most important one is context is king. And so how do you understand, how do you model, how do you control something where you don't know how it's going to act in a different context? And so with that in mind, we talked about some different aspects of modeling. So depending on what your application is, are you trying to be as predictive as possible? Are you trying to understand the emergent properties of the system? Those tend to lead you towards more data-driven or more mechanistic-driven modeling. But maybe the question for the future is, how do you bring that together to where you can use big data to essentially inform on the architecture or the structure or the properties of a mechanistic model? And maybe furthermore, how can you use mechanistic modeling to help improve the predictability of like a data-driven approach? So yeah, we were talking about at the end, like how we can put those together, how approaches like machine learning can be brought together with mechanistic models, with approaches like physics-informed machine learning, or by doing different approaches to eliminate certain models that don't appropriately fit the data, so helping to constrain the space of possibilities. So different things like that, how to put them together in a way that will ultimately lead to better prediction and hopefully enable us to look at systems and predict how they'll work, not just in the context in which they've been measured, but in contexts which have not. I guess, did that capture everything? Does anybody else want to add anything else?
That was great. Thank you, Ross. Appreciate it. Do we have brave volunteers from group 2?
So I'll just jump in. We had a great discussion in our breakout. I was doing my best to rapidly capture some of these in our sticky notes. But I think a lot of great ideas were brought up in terms of looking at some of these fail-safe manner approaches in terms of engineering cells. I think we had some of the similar interests in terms of this model-informed machine learning to connect high dimensional data into lower dimensional dynamic systems. And then we were looking at some of the limitations, again, data limitations. So I think from there's always the split between experimentalists and modelers, and understanding what is being captured in terms of the data and how can that be incorporated into your modeling approaches. And so really one of the points raised in our group was really being able to attend some of these workshops to learn, and one of the reasons why they were here today. So I think that was great. And then really going outside of our comfort zone, right? And so I'm glad you all joined in to do this Jamboards. This is how we get it, kind of some of these challenges. And then the other thing I posed to the group was, in terms of getting at some of these questions, how is training in these areas? Do we need more in terms of these hybrid, training approaches that really, before it was, we had a lot of the push on data scientists, and now things are more readily accessible in terms of your modeling, your algorithms, your libraries to be able to do some of this, but really having a grasp on the fundamentals of the biology to ask the right questions. And so we had a good discussion on that as well. And so that's kind of our summary for our group 2 in this topic 1. Did anyone in group 2 want to add to that I might have missed the finer points?
So maybe we can identify in each group what we think is like, I guess, we didn't even talk about robust control much, to be honest, because I think we were so stuck on the modeling, and really, like, if you think about methods in control theory, like they really rely on predictive models. Like all the work you see with drones, everybody has a predictive model of a drone. So it's not hard to, it's not that it's not hard, but it becomes exceptionally hard when we don't have a predictive model to rely on. And so then I can understand that probably like that might be out of the expertise of a lot of people, just trying to even figure out what such control systems should be able to do. And so I guess we are stuck on the modeling side, right, as a foundation for that. So yeah, I was thinking like maybe a good exercise to figure out like what are the major barriers or the major point of, I guess, disagreement and how to move forward in this, right? But I think that we touched upon, like, it's gonna have to be some hybrid approach where we all kind of meet in the middle. And I think what I would really like is to understand more of, you know, when I work with experimentalists, they're really excited about machine learning. They're always, and they love it. They're like, oh, can you do machine learning on this? Like, without really even knowing what that means or what we're trying to get out of it. Can we do machine learning to answer this question? Can we do machine learning to answer that? And I really appreciate that, because it will force me to think beyond the bounds that, you know, it's already hardwired in our brains when we're trained up in these fields. Like, no, that's impossible, right? But really start to get creative, and then sometimes it becomes very engineering solutions, like almost to the point, like, you know, I have to throw out any and all theory out the window and just come up with practical ad hoc solutions for everything. So that's where we are in terms of how can we facilitate more, how can we facilitate interactions between pure experimentalists and pure, like, computational people? Like, where's that gap, and how can we increase that communication to figure out what are the methods that pure computational people or theoretical people should be focusing on and are unaware of? Maybe they're focusing on the wrong things. I don't know if anybody else comment. I'm just trying to incite some conversation.
I can say something, if that's okay.
Yeah, thank you.
So I would like to talk about from what I've seen in my institution that has been successful. This has been where we have groups of people work together for over many years, maybe over 15 years, where they work on problems. So the biologists will bring problems that they find interesting, and they have these meetings weekly. And then they have mathematicians and statisticians who sit in and listen to them. And then people will just give them ideas of how to solve the problem, or even work on models with them. And then they work on joint papers together based on what they're doing, work on getting funding to work on these specific problems where they're combining modeling, mechanistic modeling, statistical tools, and then working on a particular biological problem. And I've seen this being very successful in lots of different groups, in different institutions, where you have groups of people come together and work on a common problem. I think that would help a lot. I use mechanistic modeling, and I see that sometimes you make a model based on what you read in the literature. I think that's what we used to do, most of us. A long time ago, we model, let's say, for example, cell movement, and then you read the literature, and then you make a model, a mathematical model. But then a biologist will look at it and say, well, we've done this many years ago, or maybe we knew this many years ago, or maybe what are you trying to do, or how does it apply to the problem you're trying to solve? So I think more communication across board would be very helpful. So that's what I think.
I really like your idea of having the biologist present and the computational people just giving ideas. Thank you for sharing that. So I'm told that it's about, we'll let Anya speak, and then go ahead.
Well, just something I've noticed recently, particularly since we're talking about machine learning models and things like that, I feel like there aren't a lot of statisticians that are working in biology. And to go back to your story of how, you know, experimentalists ask you, you know, can you do this using machine learning? Can you do the other thing? I feel like we're not hiring statisticians who actually understand the limits of machine learning, possibly because they're being taken away by other industries. But it's, in my experience, I don't know, kind of like, I feel like in biology, maybe you have a bunch of computational people who are trying to learn machine learning now, and sometimes we don't really have the statistical foundation.
That's really fair.
We have time to discuss, or you want to move on to the next one?
So if It's up to you. So we can hear, if you have an additional comment to add, we can, and then we'll follow with the bio break and then topic 2.
Sure. So, you asked the question, how are we gonna increase the interaction between these two groups? I mean, obviously, education is number one. Personally, I love mathematics, but I become biologist, but I'm doing both of them, but mostly experimental side. And then, but I still interested in, mathematics, so I approach to people to collaborate. But, we should do more, but more practical way to increase. Of course, the funding agency increased the course that related to, integration of mathematics and experimental one. That, if we have more core, people naturally interact and then form a team. But I do not see more. I want to see more, but we do not see many at this moment.
Yeah, I agree that maybe like we need to pose problems that cannot be solved without the communication and integration, because I think if we can find a way to work independently, we will, without really being forced to talk to each other every step of the way in the experimental and the computational side, and being forced to understand what the other group is doing.
Yeah, absolutely.
That is really needs to come out.
Yep.
Okay, so with that we'll wrap up topic 1. Let's take a 5-minute bio break. So we'll resume at 10:25. And then we'll do hopefully mixed up breakout rooms again and continue to topic 2. Thank you.
Okay. So you guys all have the link to the topic 2 Jamboard. We are breakout group 1. Okay, so, and like I was saying before, let's try to see if we can just get all our ideas down, and then we can try to, as a group, maybe flesh some of these out. So I want to take just a few minutes for all of you just to go on to the board and just put down your rough thoughts. So in terms of what is your latest and greatest in this kind of under this topic 2 prompt, what kind of solutions can we look to for mathematics for? What field can be most benefit sensing technology? What technology is needed for intervention? So take a couple minutes, and then we'll circle back and kind of discuss what we put on this board and see if we can flesh or add to some of these. Sound good? Peter, you have a question?
Yeah, just a quick question on the word of the intervention. Is that intended more in a medical context, in a more general, like doing something to a biological system in a timely fashion based on the information?
Yeah, I think all of the above. So whether or not you want to put in a context patients, or you want to put it in terms of probing the system, yeah, I think we're looking for what would be needed to really kind of dive into that.
Thanks.
Thanks for the question. All right, so just put your notes down and we'll start discussion at 1:35. Okay, why don't we get started with discussion? So this is great. So under the box here, what is the latest and greatest in sensing and actuation? Would anybody like to jump in and talk about what they put down here? And then we can move through these boxes we have here. Maybe I'll just jump in. What really caught my attention was this one in the middle here. And maybe if anybody on the call could answer this question. So someone put rabbit switches as sensors. Unsure if anyone has used it as actuators. Curious to know if anybody on this call has seen it used in that context. And if anybody posted that would like to explain, that'd be great.
This one was mine. I just know my previous colleagues use riboswitches all the time for detection of something. They were looking at analytes in sweat or in spit or bodily fluids. But when I saw actuation, it's like, I don't know if people have used it for actuation. I know sensing, it's pretty much all over the news.
Great, thanks for that. Anybody want to add to that or discuss something they put in this box?
Well, when I think of ribose switches, I maybe naively think of them as having actuation kind of built into their function, in that whatever they're switching on, whether they're being used to regulate translation, for example, that there's the sort of sensing and actuation. I tend to think of those as coupled together.
Right. I was thinking, can they use a ribose switch for something unintended than turning the regulation on? Because we want to modify it. So yeah.
Got it.
Interesting.
I could add one thing. I don't remember exactly who did that one, but I saw something. Basically, some microbes sense some signal that is the biomarker for the disease, and then only when disease is detected, they producing something for the potential cure. And then the question that is also related to other topic, that is sufficiently fast enough to do that sensing and actuation, or that's too late, because biological systems typically rely on transcription and translation, whether that 20-minute or 1 hours response time is sufficiently fast enough to do whatever thing. In that sense, just giving actuation only, like SynLogic develop, probiotic that basically degrade the phenylalanine to treat the PKU, whether that's sufficient enough, or we need to have the sensor that detecting phenylalanine and then respond to phenylalanine. In that way, you reduce phenylalanine level only when phenylalanine is high enough. In theory, that's the more useful or better, because phenylalanine is the essential amino acid. So degrading too much also bad, because people or patients, especially baby, cannot grow without the phenylalanine. So theoretically, that's a much better approach, but do we have the technology right now? That's question mark.
Right. So one question there would be, if you have it actuated, does it switch on fast enough in the presence of phenylalanine to provide the right therapeutic effect? Or is it just expedient to keep it on all the time?
Which is kind of the amazing thing, right? We look at all these cellular molecular changes, and they're really good at filtering out this noise, right? And then really looking at how signals are consistent. And so how do we kind of realize that, but also be, like you said, do we leave it on for the feasibility's sake, or do we have a way that it can filter out these noise levels, but really see the true signal, what's being changed in the environment?
To add one more thing, if you include whatever output signal, color, or wireless signal, whatever signal as actuator, I actually love one publication by Tim Luce group a few years ago. They basically converting blood signal, I'm talking about blood heme signal, into wireless signal. In that way, you don't need to do any endoscopy to detect bleeding in the gut, and then basically the bacteria with the device kind of signaling wireless signal outside. In that way, you don't need to worry about endoscopy kind of detecting those bleeding, whatever. So that is another great one, and I consider that as an actuator as well, because that is giving us the whatever output that is useful for the diagnostic. But if we're thinking about curing, it's only the actuator, that is also not satisfying your definition.
That's great. And conceptually, we think about this, right? We talked about actuating rubber sensors. We talked about finality being responsive. I'm curious, sometimes we see the responses, we think of very linear. There's a response, we react, but I think by the time we realize that, we're 5 moves where we should be. And so I'm curious to know, when you think about this and this complexity of it, when you read out the signal, really things should be happening, on multi-pronged approaches to be able to pull this cell in. But it's easier conceptually to think about it as an on-off switch, actuating things, sensing things. How can we build models to help identify some of these redundancies, but also be able to take these inputs and realize that, we need to send out signals that actuate, whether it's 5-10 steps down the line, to kind of bring this, whether it's an evolution, we're trying to put guardrails on the system, or predict where it's going to take it. I'm curious to know how you all approach that, thinking between this linear kind of thinking versus this kind of multi-pronged complexity.
So I'm just wondering, listening to everybody talk, it seems like at the same time we want to react as quickly as possible, but when you do that, you might be overly sensitive to what's going on, and it might be very noisy. So I'm just wondering what we mean by fast enough in terms of sensing and actuating, because I think that's very dependent.
That depends on what kind of application you're thinking about. So if you're thinking about, those kind of fluctuating phenylalanine level and response, the response time should be very quick. If you're thinking about some chronic disease, you don't need to worry about, those kind of signals go away. You don't need to have the fast response. So for example, inflammation in the gut, you're using probiotic curing, then you need to respond within whatever hours, and then generating whatever protein or molecule that cure potentially inflammation in response to the inflammation. So that depends on application.
Go ahead,
I'd also
Ross.
add
And then I
that-
want to jump to the next box, but go ahead.
Yeah, sure. I'd also add that not every cell is going to respond exactly the same. So for example, if you are trying to, with the example of wound heal a tissue more accurately, you're going to have more than a single cell in there that's doing that. And so the actual response applied, or detection and response applied to the system, is sort of the distribution of all responses summed up across all the cells. And so this problem of noise in part can get averaged out by things like that. And we should think about, what is the overall distribution of responses that we see, and how does that lead to some sort of overall behavior? So maybe that means that it is hard, and the challenge should be on the detection, so that it doesn't take too long for the whole ensemble of cells to react. Or maybe it means that because one cell could react very quickly, and that one cell reacting might be bad, that you need to be very careful about what you decide to detect. So it's probably application specific, but I think thinking about the distribution of responses is important for conceptualizing what the right response is.
Thanks, Ross. Thank you all. And I hate to time discussion, but I want to, for the time remaining, I want to move through our next sections here, and then we can kind of wrap up. So looking at what kind of solutions can we look for to mathematics for. So does anybody want to jump in on this one? I think we talked about it a little bit. I see here linear sensing, bimodal or ultrasensitive, feed forward.
Yeah, I guess if I could just relate it to what I said before, how we detect inputs is very important. I think that a lot of work has focused in the past on ultra-sensitive responses, which is really good for sort of, if it's above this threshold, then there's a response, and if it's below, there's not. But I think when it comes to a lot of things like inflammatory disease, and probably a lot of other applications, you want more of like a fine-tuned response, where it has some sort of negative thing is seen that you sort of in turn put out a response that offsets it. Now, I don't know enough about the specific details of a lot of diseases to give you like an exact example of that. But that's kind of what I was thinking there is building different ways, using feedback in particular as a useful architecture for this to make input sensing and output actuation be sort of linear or nonlinear, depending on what you want the system to see, or either inside the cells that are doing this sort of activity or outside of them.
Does anyone want to add to that? Thank you, Ross.
I do want to add to that a little bit, and sort of thinking of the system. I mean, everything is context dependent. We discussed this in our earlier breakout session, but also where I can see mathematics could help is, so the big question here is, how much time do we have to respond to something, right? Not everything is a, put out the fire type of situation, right? So figuring out that time in terms of, there's a note here, feedback controllers for delayed processes. So how much of a delay can the system handle? So math can help figure out that sort of buffering capacity of the system to insults before something breaks. And it's different in different cells and in different systems. Again, for some of these like disoxyphenylalanine example, I have worked on newborn screening programs. There are some conditions out there that the baby has to be treated within like 2 days after birth. Like otherwise there's irreversible damage. Others, you can wait a little bit, right? So figuring out what that sort of flexibility in the system is before you have to step in and actuate is really important.
Thanks, Tuba. So we have a few minutes left in our breakout. I'm gonna kind of bypass what field. I think we all agree these are some good choices here. If anybody has anything to add that wasn't captured, please jump in. I really want to spend the remaining time on what technology is needed for intervention. Does anybody want to jump in on this section and what they put down? So I wanted just to get some clarification. Someone put in more sensitive real-time reporters than FPs, and I'm assuming FPs are fluorescent proteins. Okay. And then I think we talked about standards. Anybody want to comment on what is meant by the standard chassis development? Building the car as we drive it?
Oh, that was me. I was talking about
okay.
like model chassis, right? So
okay.
everyone, because that's a common theme throughout all the discussion parts as well.
Thanks, Jamal.
Especially for mammalian cells, is there's not as many tame E. coli as there are, and there's just so many cell lines in the mammalian.
Peter, I know you had a question earlier about what was meant by intervention. I was curious which way you went versus clinical or more.
I did not specify. I did not constrain myself.
Okay. Okay. any final comments on any things that we posted here? And who would like to do a kind of a quick summary for this group? Takers?
I'll take a step backward by saying Ross did a good job last time.
Okay. Does that mean Ross doesn't have to go this time? He gets to hand it off to somebody else, or how do you feel, Ross?
I don't mind, but I don't think I would be the only person that would be good at talking about this.
Okay. Anybody else want to step in? Otherwise, I think Ross would do it. Okay. Yeah, thank you all for this discussion. I know we're going to be put it back in the main room, and then our last breakout will be all together. So hopefully that'll facilitate even broader discussions. Thank you for all that you put down here. If anything comes to mind later on, as you're out doing other things, we're always happy to see those ideas posted on. You can come back and add to it. And hopefully, these things will help in terms of how we're shaping our summaries for later on. So appreciate it. Thank you for the engagement today.
And thank you.
Yeah, so topic 2 is challenges in monitoring biological response in real time and effective timescales. And so, Tiam, since you had mentioned that you were had worked in or you're working in stem cell differentiation, and it reminded me of the first project that we did with the feedback control. And so we had engineered some membrane potential reporter, I guess they're like proteins that are expressed on the cell membrane, and they read out the membrane potential. And these were on pluripotent stem cells. And we were trying to essentially direct what that cell differentiated into by regulating the memory potential. So we were trying to do some patterning. Really it was a 1 year, one of those a 1.5 year seedling funds from DARPA, which then led to the wound healing project. But I was sad that we didn't get to continue on that specific project. But I guess the point of bringing it up was that that we had a lot of timescale issues, because it wasn't the question is what is the delay or the lag between when you see a pathway upregulated, right, or memory potential, which happens at even faster timescale, and by the time you see a cell differentiating, and when we realize that okay, these cells differentiate over the course of days to a week. It's how are you supposed to successfully apply feedback control to something that it takes that long to see the response if you apply any type of stimulation? And if you continue to apply different stimulations, you don't know what the cell is actually responding to, on the outcome, on the other end. It's just so convoluted, the information that's being received and what we actually visually see in the response of the system. And so I think that's a good segue into, why it's such a challenge when we cannot serve, in real time and appropriate timescales, the response of the system and the predictive outcomes, right?
Yeah, it's so true. And even worse than that is the cells not necessarily responds to the signals, but they might do something weird. For example, have some adaptive measurements, or actually responds to rate of change of the signals, etc., and makes it even harder. So you're not sure which input, when you provide that T0, what effect it would have downstream at day 7, right? Makes it hard. I think pluripotent stem cells, the good thing is the whole, at least early gastrulation events happen in the course of maybe 40 hours or so. But when you pass that, for example, if you go to ectoderm and skin, then you're really thinking about a longer term differentiation.
So I'm adding, so I'm on the, hopefully you guys are on the Jamboard. Let me know if you need the link. So we're in breakout group 2, and I added sticky note, mapping early signals to long-term response is maybe like, what kind of solutions can we look to mathematics for, right? So just thinking on the, I don't know if that's like a modeling problem, or are there ways to engineer, I guess, are there ways to engineer sensors that give us intermediate timescale information?
Yeah, that's what I was also thinking about, in the sense that sensors are things that can help us. Because I think right now we have people using sensors where, let's say, for example, one of my colleagues here has developed a sensor that can measure how much air you're breathing in and breathing out. That's through respiration. So you put it on the chest, and then it can tell by looking at how the diaphragm is expanding, and then they can capture the breathing of how much air you're taking in. And I think that was because of with COVID, where sometimes one wasn't sure how much air, like the breathing. So that was one of his motivations for doing that. So I think if we combine multiple things, we may be able to capture some of those information, things like looking at sensors, and then maybe looking at how can we bring all the things in, let's say, mechanistic models can do something. Okay, what can, based on the question you're trying to answer, how do you take that in, add machine learning, add sensors? How can we take all these different things to combine them to help us? But then at that point, we have to know, what is it, where can we put each of these? Because they're all important at different levels.
Thanks. I added, we need to get creative about what can serve as a sensor. And combine information using data-driven methods. And basically, like, we want sensors at various spatial temporal scales, right? So on the materials side, Lee, since you mentioned your on materials, is there anything in the biomaterials side that technology that maybe hasn't been applied towards sensing and actuation that has potential and is unexplored?
I don't know much about the different technologies for building sensors, so I'm just not going to say much about that. But one thing I know about is maybe I should let someone else answer that question before I say something else.
It was actually like I was trying to include Lee in the conversation, since he said my background is biomaterials, so I was curious as to where that might intersect in this discussion. But maybe you're less motivated to interact with your audio issues. I also see Brian is new to me. Brian, if you're there, would you like to introduce yourself?
Yeah, sure. So I am Brian. I'm from Columbia University. So I'm actually a grad student here. So I'm just listening to the discussion. So I work in Virginia Cornish lab, and well, my main focus is like engineering probiotics
Oh, sorry, your audio is I can only hear like a fraction of what you're saying. I think your audio is.
Oh, sorry.
Oh, is it rubbing up against your sweater?
Yeah, yeah,
okay.
I see. Okay, is it better?
Yeah, I can repeat everything.
Okay. Okay, so I'm Brian. I'm from Columbia University, and now I work in Virginia Cornish lab. So well, my main focus is engineering probiotics and using it as vaccines and therapeutics. So yeah, so I'm here, just listening to ideas, because that's a little bit out of my field, but that's something that I want to, go towards, because like I want more control of the delivery system. So yeah.
Okay, that's interesting. So because we're right now we're on the sensing and actuation version or subtopic. So I guess what you're offering is sort of an actuation mechanism, right? So it's an intervention strategy, right? So you're delivering probiotics. You deliver that what through like a needle or through vein?
No, no, it's
Pill?
oral.
Oral?
Yeah, through a pill.
Like
Yeah,
the gut?
yeah.
And so I guess the related question might be like, how much are you delivering? What concentrations, at what timing, the frequency of the pill intake? Is it daily? Is it 5 in the morning, nothing at night? Because I guess that's what we think about in the feedback controls. Like, what is the treatment strategy, right?
Yeah, exactly. And because like I'm working on vaccinology, so like, how is that interacting with the immune system? Is there any feedback loop that you can engineer in that kind? Like, for example, like can I tune up when like the immune system produces IgA that directly targets the payload that we're delivering? So, you know, is there any things that we can control like precise? Also, like for microbiome, every person is different. Like, so like a mechanistic control might not be sufficient for that. Like, is there any kind of like say like feedback control that we can do?
So then my question to you would be, what would you need to measure to understand whether what you're providing is working or not? And how effective?
Well,
what are
yeah.
your readouts? I think that's the conversation here, is like, what are the readouts, and do we have the readouts that we need, or what readouts would be useful if we can get them?
Yeah, well, the ultimate readout is the immunogenicity, like how much you know, antibodies you produce. But you know, there might be other kinds of readouts. I mean, the most straightforward readout is just like you can measure like IgA titers over time.
Well, what is that? Like, you do blood work? Like, if
Yeah, blood work.
be taking it, they will go in for blood work.
Yeah.
And my guess is you can't do blood work that frequently because you develop scar tissue and stuff like that, right?
Yeah, that's right. Like if there's some kind of like devices that can, you know, monitor it over time, that would be good.
So it's fair to say that, you know, I guess for medical applications, blood work is a common that you look for metabolomics and proteomics.
Yeah, I mean, yes. For vaccinology, like
but we can't do this frequently, right? So need other measures.
Yeah, I mean, like how frequent do you need it? I mean, for my application, it might be just you know, once or twice per week. So that might be okay.
Yeah. Well, I don't know. There was a time where I was having to do like blood work every week, and that was like
Yeah, that's right. I mean, for you know, the current application, you know, that might be the best solution.
So, I mean, does anybody? Sorry, I'm gonna read these.
You have some nice comment from Lee on.
Yeah, I was gonna. Yeah. Oh yeah, I was gonna address that. Thank you for bringing it to my attention. Oh, Eric might be better poised to answer, but there's certainly some technologies in development, microfluidics, 3D bioprinted systems, to understand more underlying mechanisms and responses to different biological stimuli. It's interesting to think about the role these in vitro engineered systems play in providing an in-between model system. I think that brings up another good point, because, you know, in how much can we translate the information that we get from in vitro studies to actual like medical applications, right? Like to, if you're going to move to animal models. So we're doing like, we started also with like in vitro studies, single cell systems and microfluidics, but we never really found the link of how that informed our treatment strategies and wound healing when we moved to mice, and now that we're moving to pig, and trying to find the overlap and what things will remain consistent and what things will change.
Yeah, maybe I can add to this in the context that I work on, which is on multicellular systems. Is we're really learning more and more that we need to distinguish between single cell responses and the collective responses, and they're not necessarily following the same logic because there are multiple compensatory, basically like a mechanism that send feedback to system to keep it on track, even though while we are perturbing the system.
So you're saying like even just like if we're looking at a culture of one cell type, even then like it gets complicated once you start to.
Actually, I'm saying like in my own project, I'm use biofabrication micro patterns to confine stem cells, and I see difference when you have 3 stem cells or 10 stem cells, and that's the level that I'm talking about. So we get more feedbacks when the number grow, even at that scale. So
What do you mean by more feedback?
So I mean, for example, there are some mechanisms that are not active when you have fewer stem cells, but when you go, some collective behavior emerges out of this interaction and dynamic between the systems that makes everything complicated.
Right. So there's some maybe cell-to-cell signaling or something that's.
Yes, and cell-to-cell signaling, the problem with that is they have 2 ways of cell signaling. One is on track, and it is when, for example, you have a system in vivo, for example, that develops, do something. But when you put it out of the context, it also behaves somehow differently because it has some compensatory mechanisms that tries to give negative feedback to keep everything at the same level. For example, in my example, that you don't necessarily see them when you're studying a system and its natural environment.
Right. And I guess that kind of ties back to the topic one of like the predictive models, right? Not being able to come up with predictive models.
True.
So maybe we can talk about like just timescales for intervention strategies. I mean, just like theoretically, like what are the timescales that are absolutely essential to be effective in a feedback strategy on the system?
I would say it depends on, and before I continue, remember to put the technologies on the Jamboard, the technologies in the chat. Don't forget to put it on the Jamboard. Did you understand me? Like the microfluidic 3D bioprinting, you needed to put it on the latest and greatest in sensing and actuations.
Thank you.
Okay.
That was directed to Lee, right? To put it in what you put in the chat room, if you can put on the
Yeah. So to answer your question, I think it depends on the question you're trying to answer, in the sense that I'm going to bring this back to what what all of us are going to relate to. If you look at temperature patterns daily, or maybe you look at rainfall patterns, you're going to see vibrations, daily vibrations. And it was going to be like a messy data, and it would not make any sense because they're just vibrations. But if you zoom out and look at it over a year, you're going to see the trail of going up and down of either rainfall or looking at things like the temperature. So I think that there's a way you can zoom in, it looks like noise. And then there's a way that you can zoom out and then you can see something. So if you're looking at trying to understand temperature patterns, how they change over the years, you don't want to zoom in so much. You want to be able to zoom out enough for you to be able to see the pattern. So I think if we come back to what we're trying to do, we want to be able to zoom in just enough for the system that we are modeling so that we can get valuable information and not have lots of noise. At least noise based on the scale, because there's always noise every time when we collect data, but you want to be able to minimize the noise based on you looking too much into the time scale, I think.
So like I agree with that. So basically timescales that are relevant, like on par with the dynamics of the system that you're trying to study or the behavior you're trying to understand. So basically, having something on a faster time scale is unnecessary, but if we had it, I mean, would it still be useful?
I think if we don't know much about the system, then more is not, it's always a good thing. The more information you get is always a good thing. But if we are constrained, I think at some point, what you need to use will depend on what you're trying to do. So in some cases, you may need faster timescale. In some cases, you may need lower. And if you're trying to look, I'm trying to think of when you would need both. Yeah, I'm trying to see it. So let me go back to temperature and see if this works. We're looking at temperature. We have daily vibrations every day. But then if we look at, sometimes we said, okay, this is the hottest it has been in this particular day. So that's us using that day's temperature. And that's relevant because we are comparing with a day. But then if we zoom out, we could say, okay, across the years, we have never seen a temperature go this high. So I think it's always going to depend on what question we are going to answer. It's going to depend on what resolution that we should go to. But it could also be interesting if we could build a system that allows us to be able to zoom in, zoom out, by combining lots of different tools, will allow us to be able to control across different time scales.
Yeah, I want to double down on this system-specific point of view and tying it back to synthetic biology, even based on the organism that we are studying. For example, if you're looking at bacterial cells or versus we are looking at, for example, mammalian cells, the time scale that they really respond is very different. And also, for example, looking at mammalian cells, the other side of things is sometimes they have memory, and what happens to them at short time scales would have long-term consequences. For example, in differentiation of blood, we know that what happens at day 7 can really do something to what happened at day 20, 25. And they start to bifurcate in terms of dynamics. And although the fluctuations are short-term in terms of time scale, but they have long-term effects because they can somehow alter the gene regular network of a system or whatever. Probably this is less happening in bacterial cells because the time scale that they're really adapted to is not at the matter of weeks or days. So I think really depends on the system. So.
That's really, yeah, that's interesting because that reminds me of, I did a little bit of domain applications with embryonic development, and the fact that, like, you know, the first 24-hour period dictates the entire like anatomy of the fly, right?
Yeah, exactly.
Any mishaps in the beginning screws up everything for the rest of the trajectory.
Exactly.
I think that's a good thing to add to the notes. So looks like our breakout is ending in 28 seconds. If there's any other things to add on the board, go ahead. I was just trying to annotate what you had mentioned, but it won't let me because of the breakout window. Oh, there we go. So dynamics on short time scales affect system behavior on longer time scales. So then that's not even a sensing problem anymore. I mean, the sensing problem is still beyond. All right, welcome back, everyone. We had the surprise switcheroo there. Worked out. Right, so let's summarize. Let's go ahead and jump into summarizing the outputs of each of the discussion groups. So let's start in numerical order. Group 1. Is there any volunteers for group 1? I can't volunteer for you since I wasn't there. And Ross already went.
We were having that debate. So I'll just jump in.
Sorry,
unless there's any other takers of
group
group.
2 for a second. I was writing.
Oh, yeah.
My bad. Yeah, I can go. Oh, 1 second.
Okay.
I was writing down notes for myself. Okay. So yeah, we talked about some various things here, but I'll touch on some of the things that we talked about, maybe the most in depth. Sort of we started with talking about different tools that there are for sensing and detection, or sensing and actuation, so the lower left box. And there's been a lot of progress on making different kinds of tools, but I guess a need that we identified and talked a bit about, especially when it comes to certain diseases like phenylketonuria, is having more real-time sensing and actuation. So when there is some sort of phenotype or negative thing that is observed, being able to quickly respond to that. And that led us into some discussion of how sensitive do you need to be? How quickly do you need to react to things? It kind of then goes into, okay, well, are you looking at a single cell? Are you looking at ensembles of cells? What is the particular application? Because when you're thinking about having the ability to real-time control a system, if you overreact to every little input, it can be bad if every single cell needs to react a certain way. But maybe if you're looking at sort of controlling the behavior of a whole tissue, if each cell is doing something slightly different, but it averages out to be okay, that might be all right. But yeah, so the ensemble behavior, the timing, the sensitivity are all very important, especially like when we start talking about using things like feedback control. Feedback inherently has some delay to how it works. And so in some cases, you want that delay to be as short as possible so that you can have a fast response. And in other cases, you might actually want to purposely build in a delay in order to get to wait or filter out things that you don't want, or just because from the time that some input is seen, you actually want some delay in intervening in that response. So yeah, those are some of the things that we thought were interesting. I think I might be missing something as part of the discussion. So if anybody wants to jump in and add anything to that, go for it.
Thank you, Ross. Any others from group one?
All right. Well, you'll have your chance after group two summarizes too. I'll give anybody in group two an opportunity to speak up, but if not, I'll go ahead and summarize. Going once, going twice. All right. So we mainly focus on two topics. I think one was just talking about timescales, like what are the timescales that are important to look at. And I think that some people brought up the good point that the timescales that we need for feedback really depend on the timescale of the relevant dynamics that we're looking at. But if we don't know much about a system, then a finer resolved information would be better. But then another group member brought up the good point that in the complexity of biological systems, it's not necessarily true that the information that we're trying to receive about the response of a system, the timescale of the response is not necessarily always directly related to the timescale of the input of the system. So sorry, that didn't make much sense. But I guess what I was trying to say is that there are dynamics that can happen at a really fast timescale early on in a process that can affect the long-term response of the system. So then the long-term response of the system is on a much longer timescale than is the stimulus, which leads to that. So because of that disconnect, you may not even, looking on something on the order of days for response to the system, you may have to look at something that's on the order of minutes a week earlier than you expect to see the response. And so one of the examples was either in stem cell differentiation and embryonic development and other applications where there's this huge disparity. And not just that, but there's also like the long delay in the response, which I think group one had brought up. And so how do you infer that mapping about what you did what you do on day one, how that affects your system response on day 7, for example. And so I'm glad that you guys had a little bit more expertise on the technology side, because I think we were a little bit lacking on what are the existing, what's the latest and greatest in the sensing and actuation realm. And then also for sensors, sorry, let me see. Okay, so we also had the comment that we need to also get creative about what can serve as a sensor, given that we need to incorporate and basically integrate information at various timescales because we don't know what's the timescale of the timescale. Oh, you guys can see everything. We don't know what information at what timescale is affecting our system, right? And how we can combine this information potentially using data-driven methods. So that was the summary of our group.
That was great. Something you brought up kind of reminded me of what we've been discussing in some of these groups was, like you just mentioned, the timescales, we think about the conception of this linear where we have an input, we want to react. But sometimes when we want to put guardrails on a system, whether we want to control the evolution or clone evolution, especially in the context of cancer, of identifying these cells that, find the way to be able to be the one that breaks through the barrier to lead the other cells to secondary sites. So, and it was brought up in our group is, what is fast, what is too fast? You'll be oversensitive. And realizing that sometimes with these inputs, as you mentioned, long versus short, you really have to be, once you kind of observe something, you need to be steps ahead to be able to control it and contain it, right? And versus, kind of being this whole balance between reactive, proactive, and predictive. So that was interesting.
I'm gonna say that this conversations have made me a little bit more depressed because I feel like we came in thinking like, oh, we're gonna do control, develop sensing and actuation, and then I realized like I don't know that we are at that sufficient understanding stage yet, right? Because in order to have like all these questions about that we've been bringing up, we would need to have some inclination of understanding in order to develop then the right mathematical tools, the right sensors, right? And to target the things that we know are going to be most impactful and most effective in our strategies. And right now, it seems like we haven't even answered that question, in my opinion. I don't know if anybody else has a different opinion on that. But this has been a bit eye-opening for me. But in my work, like I guess I've been doing those two things in parallel, in some sense, working on the control methods as also trying to understand, but also, but as we try to execute the control methods on real biological systems, basically this has also helped bring to light, the additional barriers and challenges that we hadn't seen before. So in essence, it was necessary to attempt in order to really understand where we are, like that frontier and the boundaries that we're trying to break through. And so I would encourage you guys to, you can go back to the Jamboard from topic 1 if additional things come to your mind. Again, like, point is to gather as much information as possible. You can go back to Jamboard 2. And Eric, shall we continue to topic 3? And I guess time does fly because it's already 11:05.
To round out our session today, and you guys have all been great. This will be our last endeavor as a group. So I'm putting this as topic 3. No breakouts. This will stay here in the main room. So go ahead and click that link, and I'm going to pull it up.
So I think this is your chance to vent out any additional items, barriers, concerns, questions, anything that hasn't been addressed that maybe you're like, oh, we should have addressed these other topics that we didn't think of. Like if you can come up with another topic discussion, what would be an important topic discussion that we weren't able to touch on? And you can put in the Jamboard, you can raise your hand, and I can fill in the sticky note as you're talking.
So let me start. So this one, basically raise very important question. The first one, animal versus single cell. But to me, animal is good enough to mimic the human. That's like so another question people are asking. So now, even scientists have been working on using animal for many, many years, now rely on more organoid with the human cell, because they now realize mouse is not sufficiently mimicking whatever the human situation. So that is probably one thing, I want to point out. And then another thing, probably not necessarily right now, but in the future for sure, that is the biocontainment. So we're talking about genetically engineered cell, including probiotic, CAR T cell, whatever. But those cells is the engineered cell, and then potentially release into the environment, or potentially have some safety issue. We have no idea. And how we're gonna deal with the safety issue and biocontainment. I wanted to bring up that 2 points here. And how we're gonna predict potential risk of those kind of engineered cell and the biocontainment consequence.
The potential risk of bioengineered cells. I want to go back to your first two comments because I'm not sure I fully understood the point that was trying to be made. So
the point is basically people study whatever disease model using mouse model. Also, people also use mouse as whatever application, kind of demonstration. But that's good enough to, let's say, mimic disease really happening in the human. And the answer is actually no. Also, another thing is people also thinking about microbiota, and then gut microbiota, skin microbiota, and then try to understand that, huge complex situation using mouse model, but the same knowledge we could transfer into the human situation. Answer is no again. So, and then now, scientists, okay, if that's not mimicking whatever human situation, what would be alternative? And then people now rely on organoid with the human cell that might be better mimicking whatever situation. So that's kind of some transition I recently observed from the community.
The part that I didn't get was that I didn't realize that it was a no on the animal not being good enough.
Animal is still good to give some idea or insight, but that is not perfectly answering whatever question, because there are many other aspects, human and the animal, especially mouse model, not that much match. But of course, mouse is cheaper than chimpanzee, whatever. And also that give a lot of data, many decades. But should we think about some alternative? And then the answer is yes.
So this is interesting because, the proposal was using organoid models, which seems even more far removed than a living animal from a human.
Yeah, so that's another problem. So NIH study section reviewers thinking mouse is much better than organoid. Yeah. But some reviewer and some people now thinking differently. So I was surprised because I thought NIH study session reviewers only thinking about mouse is better model, but most of the people are thinking mouse is better. Yeah. But that's not true.
So I've written a note where I said, where is animal work translational and for what? And the same for organoids, because I think it's domain specific. For example, like I know that we're working on mice and pigs in particular, because for skin studies, the pig is found to be the closest to the human. But that's true for skin. That doesn't mean that if you study another aspect of the biological system, right, that would translate to humans. So then it seems very specific to the research question in which is the right model or most informative model. But I think that that's important to address. An important question to ask when we do try to do translational work. So,
Curious to know if what's the most flat out impossible. I know there's nothing there yet. Does anybody have kind of this, when we say impossible, usually that, kind of translates to someone, someone's vision or goal. It seems kind of like, if I had this, I would do this, kind of my grand, would it be really exciting, but right now it feels like it's impossible.
I'm gonna be honest. I think building a mechanistic model from the bottom up for these like human physiology that's predictive, I think that's impossible. I'm just gonna say it. I don't think it can happen. We can build models, maybe they're qualitatively representative, but not in a way that can inform design in a complex system. At least that's my personal feeling, right? I could be proved wrong and that would be great.
I think to add to that, the environment that this model is in, because then you'd have to make, one in the rain, one in the snow, one in the sand. I mean, it just kind of.
Right, that's one of the elements that adds to the complexity.
But back to the earlier discussion, do we need that type of complex model? Do we need, because every single human different from another, and one model, I'm talking about governing rules and or whatever in the complex model. Can you make, even if we make the whatever model for me, explain all the physiology working for me, then not necessarily working for you. So do we need that complex model, or do we need some simple, model that giving some insight, like parameter A changing, we could have the A consequence that might be true for you and for me. I guess that's sufficient enough in terms of the model, in my opinion.
Mm-hmm. I'm adding another note to most critical to address how to address biological variability in addition to environmental. I think we've already brought up the environmental one, the context dependence.
I want to add something about the model, which I think is a nice analogy. When in mathematics we say definitely all models will break if you push them enough. And right. I think it's the same in biological models, and they're probably good for recapitulating some aspect of what is actually happening, but they're not good for other aspects.
Right, they won't generalize outside of that. Yeah, because oftentimes, like in the mathematical models, like if you push towards one end, you reach a singularity point, right? But as long as you're not at that singularity point, then things don't blow up, then it captures the dynamics.
I think something that we've circled around a little bit is kind of what we're doing in the application. So
yeah,
if we're helping to remodel a tissue, or we're helping to control inflammation, or we're helping to program development, or whatever it is, in a lot of cases, those are not necessarily just like simple set point control problems where, oh, okay, if we could only control the level of one cytokine, with this like engineered cell that we made, then it would fix the problem. And so our like notions of using feedback control and sense and response systems, I think is quite complicated actually. If you put in a feedback system to sense some dysregulated state, and then you put in an actuator to fix it, how do you design that to be sort of accurate, I guess? Like what is the control target
Yeah.
So I think that I don't know like what category that goes into here. Maybe like most critical to address is like what is the control target, and like how do you actually like transmit some information about the system to that, especially for a complicated system.
I was going to say, I love that you brought that up, because that is actually one of the main things that we've been battling in our research work. And somehow I completely overlooked it in my presentation, but just defining the control target was such a challenge. Because when you do like if you apply controls to a drone, you're like, follow this trajectory, right? It's very clear. This signal should look like that signal, and they should line up, and the error is 0, right? But it doesn't simplify to that in biology. Your outcome is like, oh, heal nicely and in half the time, or develop normally with the right anatomy structure, or abnormally if that's your goal. But to translate that to a mathematical language in a way that a controller can achieve that is like a whole other challenge. So mapping, basically, yeah, mapping the outcomes we want biologically speaking to some mathematical framework.
Or yeah, exactly. And then within that, we talked before.
Exactly.
I think it was something
within
that you
that,
mentioned we didn't quite
we
get to
talked
talk
before.
about.
I think it was
something that
which
you
was
mentioned
like
we didn't
how
quite get to
or
talk
like
about
can
in the
robust
first
control
topic,
be used
which was
without a
like
predictive
how
model?
or like can robust control be used
Yeah.
without a predictive model? Yeah, like this thing.
So-
What is the control target? Yeah, like in principle, what is the control target? Have a black box where
In
you know
principle,
what your
you
control
could
target is
have a
supposed to
black
be,
box
and then you
where
could
you
try to
know
control
what your
that control target is supposed to be,
and then you
in
can
a
try
way
to
that is
control
robust
that
to as much context as possible.
in a way
right?
that is robust to
And
as
so
much
maybe
context
if you
as
sufficiently
possible,
know
right?
what your control target
And
is,
so
and it's
maybe
simple,
if you
we
sufficiently
don't
know
need to
what
know
your
that
control
much
target is,
context,
and it's simple,
but maybe the more
we
complicated
don't need to
the
know that
target,
much
or the
context.
more complicated
But
the
maybe the
system
more
you're dealing with, the more you
complicated
need a
target,
model
or
that
the
at
more
least
complicated the
explains
system you're dealing with,
the
the more you
system that
need
you're trying
a
to
model
control.
that at least
If
explains
not, the way
the
that
system
the
that you're trying to
control,
control.
if
Can
not
be affected
the way
by
that the
system and
control
so on.
can be
And I can
affected
tell you
by
why
the
I'm
system
still
and
like
so on.
holding out for
And I can
control
tell
theory.
you
Why
as
I'm
a
still like
method.
holding out. This is really like famous example
as a
in
method.
this is really like famous example in so I took an adaptive control class, and the instructor was actually one of the adaptive control class, and the instructor was actually control dynamics, and they had researchers at Boeing adaptive flight control dynamics, and they do and he would like tell the story that always just stuck in my mind about, and he would like tell the story that always stuck in my mind about. And so they do have a model for the plane, right? But sometimes things go wrong. So they do have a model that model, right? But sometimes things go wrong, and at some point that model. And the example was that one of the ring fails to be predictive, and the example was that one of the wings, like kind of part of the wing snapped off, which completely like discount, like the model basically was useless at that point, right? It couldn't really predict what the plane would do anymore. But the adaptive controller somehow kicked in miraculously, and the plane found like some very like non-intuitive configuration or like angle in which to fly and safely land, despite the model completely failing. So somehow this the adaptation part of it just learned a new strategy just based on the information that kept coming in. Like it learned like, okay, whatever you were doing before is not working anymore. Adapt your strategy until it looks like it's behaving the way it should. And like that, so that's the element of like robust adaptive control that, would be really amazing to bring to a biological system, where again, it's as if we're trying to predict a plane with a broken wing, and that's what it feels like.
I actually have a question. Like, there are many methods in control theory where you don't need to like know the model very well, as we were just discussing. And of course, at least you and I, we know about like robust control, and whomever else has a background in control theory. Like, what is the barrier to us using this in biology? Is it just the fact that it's so computationally intensive? Is it the fact that it could only be implemented by a computer, like optogenetically or something, to cells? Like, why aren't we boxing in the context as an error and just sort of working around that?
Well, can you say more about what do you mean by boxing the context as an error?
So you know how in robust control, like basically you assume you don't know everything. So like the entire context of the cell that we were talking about and how it can screw things up, you could just like abstract that out as an error.
Yeah.
Right.
Yeah.
So then why aren't we doing this? I guess why aren't we trying to do this?
That's
At least
what
I've
I'm
never
trying
tried to do it.
That's been like a little bit of I mean, a large part of the focus of my research direction. But yeah, but part of it is, again, I think part of it is just the limited sensor information. Like an airplane has like millions of, I don't know millions, but lots of sensors on board.
Right.
And it's continuously receiving at a really fast timescale tons of information. So it can learn really quickly and respond really quickly, right? But we have nothing. We have like 5 time points and like maybe 1 condition and 3 replicates. I don't know what to do with that, you know? That's my problem.
I see.
And,
So
and
not
then I can only measure something that's far removed from the thing I'm trying to control. You know, I can see like a wound closing, but I'm supposed to be controlling like macrophage recruitment based on how I see the wound closing. Like that time scale is too far removed. It doesn't make any sense. It cannot inform me. So when you look at other things, you can look at pH, you can look at temperature. I mean, people are working on it now. We're like trying to get creative about what are other features or things we can look at to inform at the right time scales and inform the right information. So it's just a big block box too between what we're sensing and what we're trying to control. And in the airplane, that's much closer to each other. There isn't a big gap. And that's the challenge that I see from doing that direct sort of analogy, that application over to biology.
Yeah, I like that comment on the control theory. Like, and I think the way I see it, I really like where the area is going in terms of looking at the physical properties. So we look at, liquid to liquid phase transitions in cells and how that's occurring. It is hard to put a number on. I think you just mentioned, Marcel, on this macrophage recruitment versus, seeing how the system's being fluidized. And so how can we, and I guess from my background in rheology, how do you find a way to put a framework on biology that you can express it in, non-dimensional analysis, or, find ways of having these parameters that you can easily fine tune to put into these adaptive controllers. Because, it is hard. We look at, like you mentioned, the mechanism bottom from the ground up. But if you were to look at all the system as a whole and how it's changing, whether it's, state or molecular crowding, or, instead of these individual parameters or molecular changes, how can we, the framework for that to be able to plug that into control theory? Yeah, it's fascinating. I don't know the best answer.
I think that brings up another thing. Is just I'm going to put to address finding the right level of abstraction for the modeling. That is not clear. We'll abstract models based on whatever our technical skill set is. If we do reaction equations and ODs, that's what we know, then that's the scale at which we model, right? But I think we need to think more comprehensively about what is the right level of abstraction to think about the model. So for the wound healing, we have various layers. That's the highest layer of abstraction is 4 wound healing stages. So it's a 4-state system. And at the lowest level, you can model all the cells interacting with each other, like an agent-based model, right? And then there's a bunch of stuff in between. The question is, which one is the right one? Or you do a variety of them and then you combine them somehow, right? And that's a multi-scale modeling, which I know that there's been a lot of research on as well. Yeah, that's called multi-scale modeling, right? Although I haven't seen only because I recently was working on a review, I ran into some of these multi-scale models applied to actual medical applications. But before that, I had not seen a lot of it.
I want to point out I think we have 4 minutes.
Okay, so shall we then wrap it up? And I mean, if anybody has any closing statements, comments, last chance, not last chance, can put stuff in the Jamboard. And we'll have our, I guess, should we give a reminder that there's a main meeting on December 14th?
Right, I'll do that. Got a couple
okay,
closing slides. Yeah.
All right. So I'll pass it to you, Jermont. Thank you, everyone, for donating your morning/afternoon.
Yeah, so again, I've attended the 4 of them, and there've been a lot of commonality. Timescales, we've talked about, I think, at every single one. The fact that we need more information, we need more understanding. So that kind of translates to systems biology. But then again, I think there could be too much information. How do we absorb? How do we understand all the stuff that's coming at us? And then there's been discussions on reproducibility. And then, of course, the standard things, hey, we need more tools, we need tool development, and things along that nature. Let me thank again Marcella and Eric for their co-hosting duties. I think this was a good way to wrap up the 4 that we had. And let me thank the support staff and Infinity for their help here. And as you can see on the slide, this is our annual meeting. This is on the 14th of December next week. So we will hear from these invited speakers, and then they'll summarize their breakout sessions. And that's it. So again, thank you very much for your participation.
Thank you all.
Thank you.
See you next week.